Thank you very much for the invitation. I am Peter Odor and I am working here in Hungary in the Center for Ecological Research. I am a forest ecologist, but today I won't focus mainly on my research or the research of our research group, but I would like to speak about the results of a relatively large consortium, which is based on a cost project, the name Bottoms Up, and focused on multi-taxon forest biodiversity. And the leader and the heart of the project was Sabina Burrascano from the Sapienza University of Rome, and she prepared mainly the basic of these slides. The cost project is not a classical research project. You cannot employ people on that. You cannot pay for custom members for research. It is mainly for collaboration. So it's pay for workshops, data sharing, different social activities. So it's very good if the members has own IDs and own data and others for a common things. And based on the collaboration, you can upgrade a bit the level of the local studies. So it was our motivation. And another motivation is the forest policy in Europe, which if you see the different forest policy documents, biodiversity is always a keyword, a buzzword. Although sometimes I have the feeling that, but sometimes we really don't have enough knowledge concerning real forest biodiversity. If you see, for example, the forest strategy, it is stated that we have to protect the old growth and primary forest, planting many trees. Very important, the sustainable forest management for the future, and of course the monitoring. But if we see behind the statements, as we see yesterday, that we don't know exactly how much old growth forests we have. Some of them are not marked. Also the definition is sometimes not clear. We don't know what is the effect if they increase the forest cover with plantations for biodiversity. Also, when you speak about sustainable forest management of different way, we need knowledge what is the effect of different management types on forest biodiversity. And if we are speaking about monitoring, we are monitoring many structural elements, but we don't know exactly how it is connected directly to biodiversity, the richness and the composition of different organism groups. If we see the biodiversity strategy, we can see that we want to stricter the protection of all the old growth forest. Yeah, the same statements. And also if we see the future, for example, the logging activity, which is increased in Europe. Based on this review of the Joint Research Center, we can see that most of the activities which eliminate the logging residuals has a negative effect for biodiversity, especially if we eliminate coarse woody debris. Only some fine woody debris we could have a threshold that we can survive. And also if we look in the plantation, if we change the different type of lands to new plantation forest, if we are planting on grasslands, or if you are changing the recently semi-natural forest or natural forest, it has an absolutely negative effect. Practically, we have a positive effect for biodiversity, only the afforestation of some type of croplands. But it also depends which is the forest that you are planting. So, to be short, in the strategies we have many statements connected to management, protection, and different actions, but we not exactly know what is the effect of these different actions to the real biodiversity. If we see the biological biodiversity indicators, which is now legally collected based on the different forest inventories, we have 10 indicators for forest biodiversity. But from these indicators, we have only 2 which is directly connected to the species, like the common forest bird species and the threatened forest species. All of the others are landscape scaled or stand scaled structural or landscape level indicators. And we are not really tested still the effect of these indicators. And also, if we are looking forward to the forest monitoring law, if we are using mainly remote sensing analysis and some kind of inventories also focused mainly on forest structures. So the real direct biodiversity monitoring is I think, very far. So the motivation was that there are many studies in Europe connected to multi-taxon forest biodiversity and connection to forest management and forest structure and other things. So in this project, we want to invite all of the researchers and research groups who has this kind of data, and this is the chair. I was the vice chair, and we have many collaborators. And the first task was that we would like to merge our data. We would make a common platform when we have data for biodiversity, data from different organism groups, and also data from forest structure or forest management and other background factors. So we want to make a database and a platform in a European scale for these multi-taxon biodiversity studies. Based on these studies, we want to make a network for the forest monitoring sites which are already existing. We want to share the protocols or the methods of the different studies. Another part which was my personally was mainly involved is also the collection of forest manipulation experiments when the response variables was what different kind of biodiversity variables. And of course, based on this database, we want to make some analysis for the relationship between biodiversity, structure, and management, and improve and test the indicators that we already use for forest biodiversity. And of course, at the end, we wanted to make some policy recommendations based on this study. It was a relatively big consortium. We had many involving 300 scientists. And of course, the activity was mainly networking and collaboration. So we had meetings on work package level and action level. Of course, we have many online meetings because we was in the middle of the COVID during this project. It was very important. We kept training schools for students and young researchers, mainly for methodological parts. And it was also important that we have this kind of short-term scientific missions when we have the possibility that somebody from one research group go to another one and spend 1 month or a couple of weeks for a targeted work. The first goal was this platform. In that case, we not only collect IDs and metadata, we directly put together the raw data, all of the species records, tree records, everything to a very big database. It was a huge work. And now we have a database with 3,600 sampling units, all of Europe. We have 34 individual research projects or studies which put together the data, and you can see many more than 300,000 species records. How this database looks like. Of course, each of the records has a plot ID. This is the main basis, the sampling units, which on a higher level can be on stands, which are sometimes in one homogeneous stand you have more plots. And the most important was the site ID. It's a geographical region when one project is focused practically. And from these plots, we collected the standing tree data, practically DBH tree species and different tree level data. data for the lying and standing deadwood, and also on plot level, basically, species data. The inclusion category was, it's important, we collected that project which has this time structural data, and so we focused on multi-taxon biodiversity, so we collected project which studied minimum 3 taxa, including fungi or animals, so it's not a vegetation database, it's a multi-taxon database. The first task that we first describe the structure of this data and the representativity. For example, you can see there are more than 30 organism groups, but if you see which are the organism groups you have enough data for some kind of analysis, it is 3: vascular plants, bryophytes, birds, lichens, fungi, and beetles, mainly saproxylic beetles and ground beetles. If you see the representativity for different forest types, the blue bars show the area of the forest type in Europe, while the green one is the proportion in the database. You can see that researchers like very much the beech forest. So beech forests are oversampled, and also mesophytic deciduous forests. We have much less data from the Mediterranean forest, the evergreen Mediterranean forest, and also from the riparian forest, floodplains, different kind of wetlands. The main management types are quite well represented, except copies. In our website, you can filter the database, and you can see many metadata and the representativity all for this database. And of course, the first step was that we fix the conditions of the using this database. So fix what is the question of authorship and what is the rules for that you can use this database. So we try to make a win-win situation for everybody who is involved. Because we have many studies, it's also a first view that you can see the methodology. And we published a paper and also make a handbook that which are the most common methodology that the different researchers used for different inventories for different taxa, and try to generalize this methodology, and we made recommendations for a more intensive and a more extensive sampling if somebody in the future would like to do this kind of multi-taxon biodiversity research. It was another interesting thing that we had to harmonize the terms. This was mainly biodiversity data collected by ecologists and biodiversity experts, and sometimes the definitions and the description of the management of the forest types, the forest structure was a bit fuzzy. So if you see, sometimes we don't have real information from the management. And if you would like to analyze the effect of management or the effect of structure for this data, you need a more or less standard terminology that. So we have to harmonize that. It has two ways, a top-down and a bottom-up way. First, we take all of the papers which was published from the individual studies and try to extract the management descriptions. From the other way, we asked all of the participants and the custodians to describe which was the management type, which was the last intervention, and so on. And here you can see that this was the most representative management type: shelter wood forestry system, selection cutting, clear cutting, copies, and clear cutting with retention, and so on. And based on this harmonization, we can go further for the analysis part. Other methodological importance, which is also possible with this database, to see what is the necessary sampling intensity for these different organism groups for representativity. So we calculated it for species richness and species composition, studying the effect of sample size for this interpolation curve as the saturation of the species richness, and also similarly for species composition, we studied the variance of the species composition and tried to calculate what is the minimum sample size within a site, how many plots you need for different forest types and different organism groups, or within a forest type, how many sites you need to reach this kind of representativity. And you can see that it's very different for some organism groups, like fungi need much higher sampling intensity for a real good data than, for example, for birds or bryophytes. It's also, these graphs show this connection with the observed number of the sample and the necessary. So this part of the graph is undersampled for species richness or composition. This is well sampled, and you can see that if you see the organism groups, that mainly for fungi, beetles, vascular plants, and epiphytic lichens, the currently existing database is usually below the threshold of a good representativity. Another goal was a coordinated network of manipulation experiments. In these experiments, we collected the experiments when the researchers made some intervention for the forest structure, make different type of cuttings or anything, and the response variable was multitaxon biodiversity. There was about 30 experiments in Europe for this study, and here we published first which is the representative, which type of experiments we have. You can see that the main intervention was different type of cutting, thinning, gap cutting, clear cutting, retention tree cutting, and sometimes floor manipulation. And always they have, of course, a control. The control is in most cases a closed managed forest. There are many research focusing on deadwood or habitat tree manipulation. But there are much less the effect of herbivores, and there are some individual studies for burning, water manipulation, and forest floor manipulation. Of course, very difficult to generalize results from the individual experiments because all of them has different treatments and so on. But we can try to do it for the thinning and gap cutting inventories. And you can see that the zero is the closed forest, and another side is the effect of biodiversity of gap cutting and thinning. And you can see that mainly gap cutting has a positive effect for different organism groups, for vascular plants, saproxylic beetles, and fungi. For the forest understory, the vascular plant composition, we studied in this database the functional diversity and the functional redundancy using plant height, seed mass, and leaf structure as a trait, and compared the functional diversity, for example, of different forestry systems. And you can see that comparing to the zero is the unmanaged forest, so this was the reference. And comparing to unmanaged forest, the simple clear cutting and copies with standard significantly reduce the functional diversity and usually increase the functional redundancy. If we put these three factors, redundancy and diversity, functional diversity and redundancy together, and species diversity, the dominance is the opposite of the diversity, the selection cutting was very similar to the unmanaged forest, will clear cutting with retention and shelter would have a bit lower diversity and lower functional diversity. And so the clear cutting, the simple clear cutting is very far usually from this unmanaged and this part. Also, we try to do some analysis for the thresholds of different important structure variables like deadwood. So we try to define the important threshold for lying deadwood and standing deadwood for different organism groups and also for multi-diversity. And we resulted that this threshold, which when you reach a good or acceptable diversity, is about 70-60 cubic meter per hectare in the managed forest. Another important task was testing the recently existing biodiversity indicators of the EU. So these are the biodiversity indicators that we are collecting, and we test the correlation for the biodiversity of different organism groups. And you can see there are some good indicators and there are some bad or neutral indicators. The good indicators are tree species richness. 4 organism groups correlated very strongly with this one. Also, deadwood is correlated very well with birds, lichens, fungi, and bryophytes. And probably this was the 2 best. It's very interesting that sometimes vascular plants and sometimes birds has negative correlation with some of the biodiversity indicators. Sometimes vascular plants diversity respond positively to disturbance and human intensity, management intensity. And also it's not on the table, but sometimes the combination of these indicators has much better response in the models than the single ones. We have, of course, it was a 5 years old or 4 years old project, so the main results was the contribution and the building of this database. So we have many preliminary results. You can see that most of the publications still under review, and still we have very possibility to analyze this data. So I think this work is not finished, but it was very important to do some recommendation. So we made a recommendation for forest monitoring and policy paper for forest monitoring and sustainable forest biodiversity based on this individual research, and all of them is, of course, available on the website of the project. This was the core group of the project, and there was many collaborators, and I would like to thank you for your attention.
Before we move on, Peter, I just have one short question, and that is, I have the feeling sometimes there are 2 different academic worlds. The one that you have been describing is really trying to understand the ecosystem forest, and I don't really know how much we actually know about the forest ecosystem, despite the fact that it actually is the most dominant terrestrial ecosystem that we have. On the other side, you got the academia that is primarily concerned with maximizing the volume of wood that can be extracted within a given time. Can you, with your research, yesterday we also had this question, you know, I mean, science needs to speak up. And with science, it actually really means the kind of people who are doing this kind of research. Mm-hmm. Do you have the feeling that you are impacting the real world of forest management with your research?
I think yes. In my research, especially Hungary, I'm focusing mainly the effect of forest management on biodiversity with different experiments and other tasks. And I work together very intensively with foresters. Also with nature conservationists, but also with foresters. It's very important the increasing the wildlife and the protection of primary forest. But 95% of the European forests are managed, and they will be managed. In most of the European countries, or many of them, conservation is integrated to forest management. So it's a very important task to harmonize the forest management. One thing is the forestry system. It's very important, but mainly similarly important to keep enough residuals for biodiversity independently from forestry systems. And I think I am quite strong collaboration with foresters, and I have a positive feedback, but that the thinking is changing slowly. Forestry is a slow profession, but it's changing very.
Okay, I hope it takes them there faster than a tree needs to grow. You know, I mean that's 60 years about. Yeah. Okay, thank you, Mary. Quick one.
I think it's good. Yeah.
That was an astonishingly good presentation of an astonishing project. Really fantastic. I wondered if you could comment on sort of qualitatively the value of biodiversity in and of itself. You mentioned it's like mentioned 73 times in the document, and to me it seems like biodiversity has taken on this value unto itself as just a value, but that needs to be promoted at all costs. But sometimes, like you saw higher biodiversity in disturbed systems, which isn't surprising because you have a multiplicity of different habitat types. But if those kinds of systems only arise because of human intervention, is that really, like, what's the value of managing for biodiversity? Like, what's your perspective on that?
Peter, can you give a short answer?
Short, very short. You're pointing a very important question. What is the best measurable variables for biodiversity? The most simple one is species richness, but it's very often misleading. Like for vascular plants, species richness can increase, but the species are completely different. So if you really want to value biodiversity, you have to go back to the reference, the natural sites, the primary forest, and comparing that as a reference and comparing everything. Yeah, exactly. And also you have to use the species composition, the traits, and the qualitative, not only the species richness. Yeah.
Okay.